The result did not make sense at first. The sample had been processed twice, then a third time, using stricter controls. Each sequence was compared against increasingly large reference data sets, spanning thousands of modern and ancient genomes. The expectation was routine. Even degraded material, even incomplete fragments, almost always settle somewhere within known human variation. This one did not. There was no clear alignment, no consistent clustering, no stable placement alongside any defined population group. At first, it was treated as a technical failure, because results like that are not supposed to happen. Human genetic variation, as it is currently understood, forms patterns. Broad ones, subtle ones, overlapping and sometimes difficult to interpret, but patterns nonetheless. Even the most complex mixtures still fall between known populations. Even the most distant ancient individuals can be approximated, if not precisely identified. So when a genome appears to resist, classification entirely. The immediate assumption is error, contamination, degradation, mislabeled context, something had gone wrong. But the sample held. Repeated extraction produced the same irregularity. Different sequencing methods returned the same ambiguity. Statistical models refused to stabilize around any simple explanation. Each attempt to place the genome within an existing framework only exposed the limits of that framework. And gradually, the problem shifted. It was no longer about a failed test. It was about an unexpected past. To understand why this was unsettling, it is necessary to move backwards, not into the laboratory, but into the city itself. Rome has long been treated as something fixed, not politically, not culturally, but biologically. There has been an enduring assumption, often unspoken, that the people of ancient Rome were, in some broad sense, local, that they emerged from the Italian peninsula, shaped by its geography, its early societies, its gradual consolidation into a state. This assumption was never absolute. Historians have always recognized movement. Trade routes stretched across the Mediterranean, armies marched, settlers relocated, enslaved people were transported across vast distances. But these movements were often framed as additions to a core population, a center that remained, if not unchanged, then at least recognizable. The Romans, in this view, were still fundamentally Roman. Archaeology seemed to support this stability. Burial sites in and around the city revealed continuity in material culture. Pottery styles evolved, but not abruptly. Architectural forms shifted, but with recognizable lineage. Inscriptions, when they appeared, tied individuals to familiar naming conventions and linguistic patterns. Even when foreign elements were present, they appeared integrated rather than dominant. So the working model persisted. Rome expanded outward. Its people remained largely in place. Then came the genomes. The remains had been discovered across multiple sites in central Italy, some within the city itself, others in surrounding regions. They spanned a wide chronology, from prehistoric settlements through the imperial period and beyond. At first, these were studied in the traditional way, contextualized through artifacts, burial practices, and spatial relationships, assigned approximate dates, interpreted within known historical frameworks. Nothing appeared unusual. But the introduction of genome-wide ancient DNA analysis changed the nature of the question. Instead of asking how these individuals lived, researchers could begin to ask where their ancestry might lie. The expectation was modest. Some variation certainly, minor signals of migration, perhaps traces of contact with distant regions, especially during the height of the empire, but within bounds, within something that could still be called local continuity. The first results were not alarming. Earlier periods, particularly the Iron Age, showed what might be expected. A mixture of influences, yes, but anchored in the Italian peninsula. There were hints of connections to other parts of Europe, occasional signals pointing toward the Eastern Mediterranean, but nothing that disrupted the broader picture. Rome, before its rise to power, appeared genetically similar to its neighbors, complex but not anomalous. Then the timeline advanced. Samples associated with the imperial period began to accumulate, and the patterns began to shift, not abruptly, not in a way that could be immediately summarized, but gradually, through repeated analyses that refused to align with expectation. Individuals who, by archaeological context, belonged to the same period did not cluster together genetically. Some aligned more closely with populations from the Eastern Mediterranean. Others showed affinities to North Africa. A few appeared closer to regions further west or north, but these were in the minority. The variation was not incidental, it was structural. Attempts were made to simplify it. models were constructed, combining local Italian ancestry with a second source population. The idea was straightforward: a base population supplemented by incoming groups from known regions of the empire. But the models failed, not catastrophically, but persistently. No two-way mixture could adequately explain the data. Even when plausible source populations were selected, regions with known historical connections to Rome, the statistical fit remained poor. Residual signals suggested missing components, unaccounted influences. Something was not being captured. At this point, the interpretation could still be cautious. Perhaps the reference datasets were incomplete. After all, ancient DNA is unevenly distributed. Some regions are well represented, others scarcely sampled. It is entirely possible for a real historical population to be absent from the comparative framework. In such cases, genomes do not align cleanly, not because they are anomalous, but because the system used to interpret them is incomplete. But the uncertainty deepened. More sophisticated methods were introduced. Haplotype-based analyses, which track longer segments of DNA and can detect finer-scale relationships, began to reveal clusters within the imperial population. Not one cluster, several, each overlapping with different regions of the Mediterranean world. Some individuals showed strong affinities with populations from the Levant. Others aligned more closely with North African groups. Some occupied intermediate positions, suggesting complex mixtures rather than simple origins. And crucially, only a small proportion resembled earlier local populations in a straightforward way. The idea of a stable Roman genetic core became difficult to sustain. Instead, the data suggested something else. A city that had become a convergence point, not metaphorically, but biologically. At this stage, the anomaly, the sample that resisted classification, began to make more sense. It was not necessarily outside human variation, it was outside the available map. If Rome, during its imperial period, drew people from across vast and poorly sampled regions, then some individuals would inevitably fall between known reference points. Their ancestry would reflect combinations that had not yet been fully documented. In that context, a no-match result is not a contradiction, it is a gap. But this explanation is not universally accepted. There are other possibilities. Contamination remains a concern. Ancient DNA is fragile. It degrades over time, breaking into short fragments that are vulnerable to modern interference. Even small amounts of contamination can distort results, especially when the original material is limited. Laboratories use multiple safeguards to detect and account for this. Damage patterns characteristic of ancient molecules are used to distinguish authentic sequences from modern ones. Statistical methods estimate the proportion of contamination. In most cases, these controls are effective, but not infallible. There is also the issue of preservation. Some environments are more conducive to DNA survival than others. Heat, humidity, and soil chemistry can all affect the integrity of genetic material. Poor preservation can lead to incomplete data sets, which in turn can produce unstable or ambiguous results. And then there is the question of classification itself. Genetic categories are not fixed. They are constructed based on available data, shaped by the populations that have been sampled and the models used to interpret them. As new data emerges, these categories shift. Boundaries blur. Previously distinct groups begin to overlap. In this sense, the absence of a match may reflect the limitations of current frameworks rather than the uniqueness of the individual. The debate continues. Some researchers emphasize the robustness of the broader pattern. The consistent shift in ancestry during the imperial period, the repeated appearance of Eastern Mediterranean affinities, the overall increase in heterogeneity. Others urge caution. They point to the uneven distribution of comparative data, the potential for bias in sampling, the difficulty of reconstructing complex population histories from incomplete evidence. Both positions acknowledge the same underlying fact. The genetic history of Rome is not simple, and perhaps it never was. The implications extend beyond a single city. If the population of Rome at the height of its power was as diverse and dynamic as the genetic evidence suggests, then the concept of Roman itself becomes more fluid, not a fixed identity tied to a specific ancestry, but a designation that encompassed multiple origins, multiple trajectories, converging within a shared political and cultural framework. This does not invalidate earlier interpretations. Archaeology, history, and linguistics still provide essential context. They describe how people lived, how they organized themselves, how they understood their world. Genetics adds another layer, one that reveals movement, mixture, and transformation at a scale that is difficult to detect through other means. But it also introduces uncertainty, because the genetic record is incomplete, because models are approximations, because the past does not always fit neatly into the categories we create to understand it. The sample that refused to match any known population remains unresolved. It may, in time, find its place as new data becomes available. It may turn out to be less unusual than it appears now, its ambiguity a product of current limitations rather than historical reality. Or it may continue to resist classification, a reminder that even with advanced techniques, even with expanding databases, there are still aspects of human history that remain out of reach. The city of Rome, once imagined as a center defined by its continuity, now appears as something else. A point of intersection, a place where histories overlapped, where identities were layered, where the boundaries between populations were not fixed but constantly shifting. And within that complexity, it is perhaps not surprising that some traces do not align cleanly with what we expect. They were never meant to.